Good morning, everyone. Welcome to the June webinar for the Atlas of Living Australia. My name is Martin, and I run the Science and Decision Support team for the ALA. Before we begin, I'd like to start by acknowledging that we're broadcasting today from CSIRO's Black Mountain campus, which is in Canberra, and that's, of course, on Ngunnawal and Ngambri country. I'd like to extend my respects to elders past and present, and extend those respects to any Aboriginal or Torres Strait Islander peoples on the call today. So today's webinar is a very exciting one for me, and I'm sure for our speakers, and I'm sure for you at home as well. And just to give a little bit of introduction, I'm going to get out of the way as soon as I can and hand over to the floor to our delightful speakers today. But look, I don't think it's too surprising to people that technology is moving very fast at the moment. And I think things like AI get a lot of attention and a lot of headlines. But I think what gets lost along the way sometimes is that a whole set of systems of technologies are evolving equally rapidly, and including the support systems, things that enable us to store and process information. And so today's speakers are here from three different NCREST facilities, so sister facilities to the Atlas of Living Australia. And they're here to talk about some of those new technologies and how they're helping us in species description. And I'm, as someone who works at the Atlas of Living Australia, where we are more often recipients and people who share data, to see people who are generating new information and working directly with researchers is genuinely a very exciting moment. So I'm really glad to be able to introduce our speakers today. So first, I'd like to introduce Sophie and Alistair from Bioplatform. Say hi, guys.
Hi.
We've got Donald phoning in today from Australian Plant Phenomics Facility.
Hi.
And we've got Diana, who's calling in from National Imaging Facility.
Hi,
and that's the order that people will be speaking today. So like I say, I'm going to get out of the way pretty quickly and hand over to Sophie, who's our first speaker. Sophie, if you want to share your slides, that would be wonderful. Yes. And while you're doing that, I'll just put in one plug to everyone who's watching along at home. Feel free to enter your questions. We do have plenty of time for questions, but we'll tend to keep them to the end. So don't hesitate to drop them in, and we'll try and ask as many of them as we can. Sophie, over to you.
All right, thank you very much, Martin. Good morning, everyone. So as we all know, Australia is a mega diverse continent, and a large part of its biodiversity is still unknown, as well as fast disappearing, unfortunately. For the people who are not familiar with Bioplatform, we are a NCREST capability sister to ALA, and we are enabling critical infrastructure for biomolecular science. And this is all the omics and synthetic biology, and underpinned by a digital infrastructure for bioinformatics. We have been engaging actively with the research community, both nationally and internationally, to establish some national science programs and initiatives, and that catalyze research collaboration and help to build some new capabilities, as well as critical data infrastructure to address some of Australia's biggest research challenges. Our initiatives are covering three different platforms and three different themes. So the nature and environment, which we are mostly going to talk about today, primary industry and food, and human health and well-being. For these, we're actually bringing together various stakeholders to create to sort of address some of these specific challenges, for example, threatened species, or understanding the biodiversity of native plants, Australian plants. And we are working together to create some standardized guidelines and protocol, and also referential data assets. And all of this is underpinned by that digital infrastructure that help making use of that data for the day-to-day and ongoing research for Australia. I am that was just a whirlwind start. And I am now going to pass the floor to Alistair, who is going to showcase some of his work as part of one of our most recent initiatives, that is the Functional Fungi Initiative. Thank you. I'll stop sharing.
Hey, am I cooking?
Yeah, we can see that. It's not maximized, Alistair.
Ah, primo. How about meow? Is this
Same?
maximized?
No.
Oh, raspberry.
That's okay. We can see it.
Okay, I'll just give it a crack then. All right. Hey, I'm Alistair. I work on magic mushrooms. I have done for the last 3 years as a magic mushroom farmer, and then also researching them. And what I've done couldn't have happened without a whole community of people who have sent me spores, more than hundreds of spores over the last couple of years. And of course, I couldn't have done it without support from Bioplatforms Australia through the Functional Fungi Initiative, who supported me to sequence more than 100 genomes of magic mushrooms. So what are magic mushrooms? Well, if you've ever seen a fungus, you've seen reproduction in some way, shape, or form, whether it's sexual reproduction or clonal reproduction. And to help their survival, fungi produce compounds called secondary metabolites. And we humans, we weren't around when fungi evolved, so the best we can do is hypothesize what those metabolites were for. And we've come up with hypotheses like to protect themselves, they might produce compounds that are unpleasant to taste or that make something feel unwell when they eat it. They produce compounds like this stinkhorn mushroom to attract things, to spread their spores to new locations. And fungi produce compounds that take control of minds. So this is best illustrated by things like entomopathogenic fungi, co-evolved relationships where an ant infected by an Ophiocordyceps might do things that give an advantage to the fungus, like put itself above the forest floor to rain down spores on its little ant comrades below.
Alistair,
So
I'm
these
just going to interrupt and say that we're only seeing your title slide at the moment. So that's better. Yep, no, good. That's slide 3 now. That's good. Thank you.
Okay. Okay. So yeah, well, these photos down the bottom are shrooms. They're magic mushrooms, and magic mushrooms produce a compound called psilocybin, which probably evolved 20 to 100 million years ago, and it would have targeted the serotonin receptors of things like mollusks, arthropods, and nematodes. And we can only imagine what it might have been doing, whether it was to protect, attract, or to take control of those little things that were eating them. All of those are plausible hypotheses. Through a stroke of evolutionary good fortune, when a human eats a magic mushroom, psilocybin binds to our serotonin receptors and we have profound and life-changing experiences. So much so that psilocybin is declared a breakthrough treatment for the treatment of mental health disorders. So what I was doing, I took some bioplatforms data. I just wanted to see what hallucinogenic mushrooms were around the soils of Australia. So I took some bioplatforms data from the base sequencing project and started just to compare it. So this is all species of psilocybe. And what you're looking at is a splits tree. And if you're familiar with a phylogenetic tree, which constrains a dichotomous hypothesis of evolution, this is putting down every single relationship among sequenced data. So at the end of these edges or branches, you've got circles and stars. The circles are reference sequences from GenBank and the stars are sequences from soil. And in a splits tree, genetic distance is really obvious. The further things are apart from each other, the more different they are in terms of genetics. And the closer they are, the more similar they are. And when you see a whole bunch of reticulation or squares, that's usually an indication of something complex, whether it's sharing a common ancestor or sexual reproduction in some way, shape, or form. So these little stars can be comparable to reference sequences on GenBank. So here, here, and here. But these stars down here were super interesting to me because they weren't comparable to anything on GenBank. So these are just sequences of a mushroom, not the mushroom itself, that occur in Litchfield National Park and Kakadu National Park in the Northern Territory. So 100% a hallucinogenic mushroom, but we have no idea what it looks like. Maybe it looks like Psilocybe brunneocystidiata, which was described from PNG, and the person who described it thought that it was going to be sister to these mushrooms right here. So onto mushrooms that we do know what they look like. And I've worked on 2 magic mushrooms. I've worked on the one on the left is Psilocybe subaeruginosa, sometimes called subs. They're probably in your backyard right now. If you live in an area where there's frequent frosts, Psilocybe subaeruginosa is really common in our natural ecosystems, in sclerophyll forests, in rainforests, in your local schools, your universities, your playgrounds. It grows as a colonizer of wood chips. So it is everywhere. And on the right, we've got Psilocybe cubensis, or cubes, and these occur in cow manure in Queensland and New South Wales. And they're the stalwart species that was cultivated for more than 50 years by people who worked out how to grow magic mushrooms recreationally. If you've ever eaten a gourmet mushroom like an oyster mushroom, it's because people worked out how to grow cubes, whether under their beds, in their basements, in their attics, away from the police and away from their mothers. So obviously it's illegal to cultivate magic mushrooms. So bioplatforms supported me to sequence about 80 to 90 genomes of Psilocybe subaeruginosa, and I sequenced 35 genomes of Psilocybe cubensis to ask scientific questions on their origin in Australia. And with cubes, I was really interested in how cultivation has impacted their diversity. So this is a splits tree again, this time of Psilocybe cubensis, 124 species, over 350,000 differences. And I'm really sorry that this isn't in the presentation mode, and so you've got an awkward title and subtitle there. One of the first things that you can see is that the Australian population is totally different to everything under cultivation, the rest of the spectrum here. And that's just based on this genetic distance. And so that means that cultivators didn't introduce shrooms to Australia, and our genetic diversity hasn't been incorporated into those under cultivation. Then we've got, if you look more closely, this is a little bit more cryptic, but in one piece of poo in an Australian farm, you've got more genetic diversity than in entire populations of cultivated mushrooms. So over time, or over the last 50 years, the people who have cultivated these mushrooms have inbred them. They've taken a spore print, grown a mushroom from it, then spore printed from more mushrooms. And in humans, inbreeding has terrible consequences, but in mushrooms, it just causes a loss of allelic diversity. And in something like penis envy, the genomes are completely homozygous except at the genes that control mating compatibility. When we look at those genes, we can understand that all of these cultivars stem from 7 founding provenances. That's based on mating compatibility, loci diversity. So the next question is on subs, and Australia is absolutely the center of origin for Psilocybe subaeruginosa. And we have some structuring by geography, so based on the Great Dividing Range or on mountaintops or valleys. And then we've also got interesting things happening like at Geelong and Clifton Hill. So 2 distinct populations or distributions that have the same founding diversity. And that's probably because the council is moving wood chips and mulching garden beds. And it explains these Northern Hemisphere species, Psilocybe azurescens and Psilocybe cyanescens, which look like Psilocybe subaeruginosa, but are described from the Northern Hemisphere. So our mushroom has probably been introduced to the Northern Hemisphere by the movement of wood chips, mulch, or plant material. Okay, I've got a little outcome slide, which I won't tell you about, and you can just catch up. If I've piqued your interest with anything here, you can hit up my blog, check it out, and I've talked about everything there. All right, thanks a milli.
Awesome, thanks heaps for that, Alistair. I actually did not know you were going to talk about magic mushrooms, and that was fascinating. So it's funny, isn't it?
What a nice surprise for you.
Absolutely. No, and it's funny how I think you're completely right about this is where future medicines are going to come from, right? Is unexpected places of high biological diversity, and the opportunity to explore that is a really fascinating one. So like I mentioned, we normally hold our questions for the end, but feel free to keep pile some questions into the chat if you want at home. Donald, you're our next bee here.
Okay. Thanks very much indeed. Can you see that?
Yes, absolutely.
Okay, good, good. I work for the Australian Plant Phenomics Network. Until a few weeks ago, it was called the Australian Plant Phenomics Facility, but as I'll mention in a few moments, we've expanded significantly in recent months, and therefore we're going through a small name change. My role with APPN is as the data management director, so my big interest is how we pull all of the data together from our activities to support science. So APPN plays a role in the NCRIS ecosystem of research infrastructures in supporting researchers who want to phenotype plants. And phenotyping is the term, and phenomics are terms that are used to describe efforts to characterize plants and animals as they grow and as they develop. So it's really working out ways that we can provide data that relates to their visible traits, visible or biological traits. And the big goal of plant phenotyping is for any particular genome, and by that typically we're talking about crop varieties, seed cultivars for wheat or barley or any other crop plant, how that genome interacts with the environment in which it's grown and the management strategy of the farmers and breeders who may choose to add water at different stages, feed, pesticides, etc. How all of those things come together to be expressed by the plant as its phenome, the outward characteristics. And when we're growing crop plants, that really particularly boils down to questions like how well do the plants grow in drought conditions, salty soils, acid soils? Are the plants stable as they grow, or do they start to topple over when they reach a certain height? And most significantly of all, what kind of yield can we expect for them? And environment in this context includes diseases, pests and fungi that may be affecting the plants and for which there's going to be variable resistance in different genomes. Historically, this information was collected by growers and breeders going through documents like this one for the Bambara groundnut. I would have shown the wheat one, but it's much older and just a typed document. For each variety of plant that anybody wanted to characterize, they'd go through a set of fields like this, assigning values to them. And some of these are very close to the Darwin core that we use in the Atlas of Living Australia: genus, species, subspecies, collection identifiers, because we're dealing with specimens of seeds, etc., that are held in collections. But on the right-hand side, you can see some of the kinds of things that were being assessed manually for Bambara groundnut. Things like the shape of the pods. Does it end with a point? Is it fully rounded, etc.? And historically, the process was very manual. People with spreadsheets just using their own expertise to code these as numeric values. These days, we have a wide range of different imaging systems that can help us to collect data on plants as they grow at all scales, whole regions, fields, smaller plots, individual plants. And as we do this, we can process those images, we can process LiDAR point cloud data that tells us the structure of the plants in order to derive useful information of just the kinds that formerly would all have been assessed manually. So if for example, we think about the kinds of sensors that may be possible from unmanned vehicles, aerial vehicles, drones effectively. There are different kinds of drones. Here in Australia, we're mostly using rotary wing drones in agricultural systems. You can stick a wide range of different cameras and sensors on these devices, and from those collect the kind of data that's shown on the right. using feature detection to count the number of plants, using LiDAR to estimate the height of the plants as time goes on through the season, using more and more complex imaging with more and more spectral bands in order to access those bands that can help us to estimate important aspects such as the nitrogen availability for the plants. And with many crops, there are models that have been developed to estimate the yield and the biomass just from these imaging techniques. APPN, still APPF here in the middle, is now a national network with partner universities and departments of primary industry around most of the Australian growing regions, and working together to try to produce much more consistent and comparable support for researchers and breeders. This includes a network of field sites where we're using a broad range of sensing technologies to observe crops as they grow in plots. We're just investing in a new fleet of drones that will include hyperspectral imaging capabilities and LiDAR, so we can get very rich data indeed through the season of the crops as they grow. And there are also tractor-based vehicles that we can go through and image at a much closer level to the crops themselves, and that can be very useful for counting wheat heads, etc. Additionally, the APPN partners operate a number of controlled facilities, whether small cabinets in which a few plants may be grown under controlled lighting, temperature and humidity, or much larger rooms with potentially more sophisticated climate control. On the right-hand side, you can see the facility at Western Sydney University, where whole trees are grown inside canopies where the carbon dioxide and other gas exchange can be controlled. At the bottom, there's an image of an X-ray machine in Adelaide that's used for exploring the internals of wheat heads to understand just the quality of the production of different varieties in different conditions. My role, as I said, is to support the data collection, and this is complicated, but still in many ways very similar to the sort of things that ecologists are involved in around the country with their data collection. Here in this representation, we have an environment, perhaps a cabinet in which plants are grown, and we may be tracking temperature and humidity through time in that cabinet. The cabinet includes several pots, each of which may have its own controlled environment with controlled water application, so we can monitor the moisture. There may be some where fungal mycorrhizae are added to the soil and others where they're not, so that we've got control comparisons for different kinds of treatment. And the plants themselves are things that we're imaging, and from those images then measuring obvious things like height, but also estimating biomass and more complicated traits. And it's possible also to be subsetting the plants to collect more precise data at different points. So at all these scales, both in controlled environments in the field, we're trying to collect data that tells us about how these individual plants and the components of those plants are developing in the specific environment in which they're grown, so that we can produce those answers to how the genome responds to its environment. And putting all this together, as we're doing this now as a national network, the goal is to be collecting consistent data for varieties across a range of measured environments, and particularly doing so throughout the geographic regions of Australia, so that we can start thinking about better tools and analyses to support future crop development, to help to optimize the agricultural systems of this country for food security, etc., and to identify the points at which current breeds may no longer be the appropriate ones for future climatic conditions. Thank you.
Thanks, Donald. That was really fascinating. It's funny. There's a facility for the APPN one building over from us, and I've never been inside, so it's lovely to get a better insight as to the sort of work that's being done there and around the country. I've definitely got questions, but I think I'll try and hold them for the end. Diana, would you mind if we hand it over to you? Lovely, thanks so much.
Are you guys able to hear me?
Yeah, I hear you, and the slides look great too.
Hi, everyone. My name is Diana Patalwala, and I work as a facility fellow for the National Imaging Facility. And I would like to thank ALA today for providing me with an opportunity to talk about NIF and how it's facilitating a better understanding of Australian biodiversity by providing our researchers open access to some cutting-edge imaging infrastructure and expertise. So what is NIF? The National Imaging Facility is Australia's advanced imaging network. We are a group of 60 imaging experts spread across some 105 imaging platforms across 14 nodes within Australia. Our vision is to enable Australian imaging science to unlock solutions to major challenges by providing open access to flagship imaging equipment, expertise, tools, data, and analysis. NIF enables research translation, right from small animal imaging involving MRI, PET, SPECT, CT imaging through to large animal up to human imaging. We facilitate the entire pipeline along with radiochemistry, data, and AI support. What NIF also does is materials imaging, predominantly X-ray computed tomography. We use CT imaging to investigate additive manufactured materials, medical implants in paleontology, fossil studies, archaeology artifacts, museum specimens, plant imaging, etc. NIF's program has 5 central goals, 2 of which I would like to particularly mention today, one being the preclinical and frontier imaging. So along with small animal and large animal imaging facilities, we are expanding our capabilities and expertise to cater to new imaging applications, such as imaging of Australian flora and fauna. The second one being imaging data collections and partnerships. We want to create and manage priority national data assets. Australian biodiversity data could be one such asset. Keeping these 2 goals in mind, we at NIF's West Australian node, which is located at the University of Western Australia, have recently acquired a large field of view materials research dedicated CT scanner. Before I talk about it, I'm just going to quickly mention here that we do host a range of preclinical imaging facilities involving CT, MRI, ultrasound, and optical imaging, along with human imaging capabilities. In terms of materials and preclinical imaging facilities, our model basically involves user trainings, one-on-one user trainings, involving instrument operations as well as analysis softwares. And we also provide fee-for-service options. In regards to the large field of view materials research dedicated CT scanner, we have the Nikon XT-Edge 225ST CT. The default field of view for the scanner is 25 centimeters by 25 centimeters by 25 centimeters, but the height could be extended up to 40 centimeters with a tall sample scanning mode. The maximum weight that the system can carry is about 50 kgs. So 50 kgs is the limit for the maximum sample it could have. The X-ray energies range from 30 kilovolts up to about 225 kilovolts of X-ray energy. This is one of the most high-powered X-ray sources in WA with 450 watts of X-ray power, max X-ray power. Resolution ranges from anything less than 1 micron to up to 130 microns. And a helical CT scanning mode is also available on the system, which predominantly reduces the regular artifacts that are usually observed with laboratory X-ray sources. So the very first project that I'm going to talk about today is the high rates of erosion on a wave-exposed fringing coral reef. This project was led by Damien Thompson from CSIRO, and it was a joint collaboration between Ningaloo Outlook, CSIRO, and Woodside Energy. So what Damien did over here was he was investigating the bioerosion of coral blocks which were deployed along the Ningaloo coast here in Western Australia. Damien and his team deployed some 51 blocks of coral species across 2 habitats within the Ningaloo region. One of the habitats was the wave-exposed reef slopes, and the other one was the lagoonal habitats. Within each habitat, they determined a few locations, and per location, 3 coral blocks were deployed. So what these guys did was they CT scanned these coral blocks pre-deployment. Then they deployed these blocks for a period of 12 months. Some of them were deployed for a period of 20 months. And at the end of the deployment period, they were retrieved and they rescanned these using CT imaging. They're trying to quantify here the bioerosion of these coral blocks by 2 methods specifically. Direct measurements, which basically involves CT imaging and scanning electron microscopy, and indirect measurements in terms of, for example, parrotfish bioerosion rates and urchin bioerosion rates. We basically come into play in regards to the computed tomography imaging aspect here at the WNIF node. So what Damien did was he scanned these coral blocks using our CT scanner here at the WNIF node, pre and post deployment. These blocks were scanned at 90 kilovolts of X-ray energy at around 35 micron resolution. 3D registration of pre and post deployment scans was done using Brooker's data viewer software, and he used Brooker's CTN analysis software to quantify the volume of erosion. Now, as you can see from the results obtained, the external erosion rates along with the total erosion rates are significantly higher in the wave-exposed reef slope habitats as opposed to the lagoonal habitats. And the internal erosion is significantly less as compared to the external erosion rates. In terms of indirect estimates as well, you could see that the total erosion rates are significantly higher in the wave-exposed reef slope habitats as opposed to the lagoonal habitats. Overall, this study provides first estimates of erosion rates across multiple reef habitats at the World Heritage listed Ningaloo Reef, with direct erosion rates of the blocks after 20 months representing one of the highest measured erosion rates on experimental coral substrates to date. So from coral imaging to Australian marsupials, my next project, I'm going to talk about comparative 3-dimensional jaw muscle anatomy of marsupial carnivores from the Dasyurus species, which would be the western quolls and the termite-eating numbats. This project was led by Vanessa Thompson from Natalie Warburton's group at Murdoch University. Now, the Myrmecobidae taxonomic order is comprising of basically 4 families, of which the Myrmecobidae is the animals within the Myrmecobidae family, and their muscle anatomy is relatively poorly known as opposed to the other 3 families within the order. So what Vanessa did was she basically scanned western quolls, which belong to the Dasyuridae family, and the numbats, which belong to the Myrmecobidae family. She scanned the heads of these specimens, pre-staining, post-staining. She basically used diffusible iodine contrast-enhanced imaging. So she used Lugol's stain, and she scanned these specimens. The energy ranges for scanning these specimens ranged from 112 kilovolts to up to about 180 kilovolts, depending on the size of the heads. The resolution ranges were in between 30 and 63 microns. And the time these specimens were stained for would obviously depend on the size of the specimens. For the smaller specimens, some of them were stained for around 7 weeks. The larger specimens, they went up to about 10 weeks within the iodine, the Lugol's stain solution. Registration of pre and post stain scans was done using Thermo Fisher Scientific's Aviso software. Of the results obtained, some of the noticeable facts were, in contrast to the western quolls, the jaw muscles of the numbats were greatly reduced. And the digastric muscles within the numbats comprised only of a single large anterior belly, while the posterior belly was absent, as could be seen in the western quolls. Now, the muscle descriptions and 3-dimensional models provided here in this study aim to further facilitate analysis of the musculoskeletal adaptation and evolution within the Dasyuromorphia order. One of the other projects that was carried out by James Barr and Catherine Bioswar from Curtin University involved ontogeny and caudal autotomy fracture planes in large skin-set lizard, Egernia kingi. So these skinks, basically, it's believed that the juvenile skinks use caudal autotomy, which is the ability to self-amputate a portion of their tail as an effective but costly survival strategy. Now, it was believed that they lose this ability as they mature, as they grow. But as can be seen over here, James scanned a number of skinks in different age-specific criteria. And as could be seen over here, the adult skink also retains these caudal autotomy fracture planes, as could be seen in the juvenile skinks over here. And they very much still have the ability to self-amputate a portion of their tail if needed, but they don't do that as much because of the mature strength and that they develop with age, basically. Then we have Dr. Alice Clement from Flinders University studying brain evolution in early lungfishes. So Alice is looking at the shape of the internal parts of the skull, the cranial endocast, to examine brain evolution in early lungfishes. She's also using some finite element analysis in investigating the feeding biomechanics in fossil lungfishes. So that's pretty much it from me. I would like to thank all our partners and all the researchers for allowing me to present their data. Thank you.
Thank you, Diana. That's fascinating to see the work that's going on through there and the diversity of that as well. Look, I'm an ecologist by training, so the lizard stuff resonates with me more, but it's incredible to see all of that coming together in one place. We've got 15 minutes or so for some conversation, which is fantastic. That doesn't always happen. And look, just coming to this as an external person, perhaps I'll lead with a question and then go to the set that are being provided by people at home. And I guess it's that across the 3-4 talks there, some of this technology is brand new, right? And quite incredible in its capabilities. I wonder if any of you have any insights you could share on the challenges of keeping up to date in such a technologically advanced space. Sophie, perhaps with bioplatforms, I mean, what are your thoughts there?
This is an eternal problem with some of these technology, mostly emergent technology, just keep changing in the genomic sphere and all of the omics sphere. The turnover of all those platforms is pretty much, I wouldn't say monthly, but definitely 6 monthly sometime. And we are talking about pieces of equipment which are very costly. So through the way we are trying to approach this is obviously our facilities. We have about 17 different facilities within our network, and they each have some specialty. So they each also sort of keep up to date to the most state-of-the-art type of things. But it is always a race to just keep on top of the state-of-the-art machines, and I suppose it's the same in all fields, really.
I see that. Donald, if I could hand to you, I mean, you're acting as a data manager and presumably new data forms are coming in at all times. Does that create specific challenges?
To some degree, I mean, new data formats tend to be larger than old ones if we want to be dealing with point clouds and hyperspec. I think the bigger challenge I see is determining when to shift to new technologies, because we want to have good time series data and the ability to compare how things are grown today with how they were growing 5 years ago. And if all of the image resolutions have changed and we're using completely different equipment and techniques, that gets difficult. So addressing the challenge of cross-calibrating different methods is, I think, something that's also going to be particularly important for us going forward. But generally, the size of the image objects that we receive does continue to grow, and that is a challenge not only for storage but also for simply processing them to extract the information we want.
Yeah, I can see that. Donna, it strikes me that you might have a different challenge, which is that you have, I mean, an X-ray is useful for humans as well. So you have many competing interests here, plus trying to keep up to date. Do those pose particular challenges for you guys?
I guess the idea here is to extract maximum capabilities out of the existing infrastructure and, create those impact stories so that NCRIS can find the value in what you're doing and keep investing your facilities, basically. So, and the added benefit, of course, of being a part of the larger infrastructure that's spread across Australia is that we can, it's basically open access, right? So we can have our users accessing other nodes facilities, and it's a cross-collaborative effort. So we are all in contact with each other and we share expertise across the nodes. So it's always beneficial that way.
No worries. Alistair, I've been given a very specific question for you about here. So obviously you've been talking particularly about mushrooms that have particular properties, but of course mushrooms are an enormously diverse group and one that's famously difficult to recognize. Do you have any recommendations for people, not necessarily looking for magic mushrooms, but just to identify mushrooms in general? Like, are the resources out there for people?
Well, sorry, I'm going to answer how to identify magic mushrooms. They're really, really easy to identify because they produce psilocybin, and psilocybin metabolizes into psilocin, which oligomerizes into a blue staining compound. So they stain blue, and then a lot of mushrooms, you can identify them based on the color of their spore print, and psilocybe produces a purpley brown spore print, and they have gills, whereas other fungi, a spore print is always good within the group of mushrooms, the Agaricomycotina. So spore prints, but I mean, me, mycologist or former mycologist, I would always be using DNA sequence data. Just because microorganisms, humans are applying our little concepts of a species to microorganisms, and we barely even understand how they speciate. So getting a DNA sequence is always a sure thing.
So presumably for citizen scientists, you'd suggest they were somewhat careful in the field.
Well, I mean, if you want to learn how to identify mushrooms in general, then gosh, the citizen scientists are the ones driving the research, because there's not really that many people researching mushrooms in our scientific community. There's not any funding for them. So you can join a mycological society up here in Queensland. You've got QMS, Queensland Mycological Society, and you can learn more from this community of people than I could ever teach you. That's for true.
No, it's a fair point, actually, and we find the same. It's a real privilege at the Atlas of Living Australia to be able to store and retransmit that data to researchers, and there's some incredible insights that are being derived from citizen scientists in many different aspects of biodiversity. Donald, I wonder if I could go back. We've had a few questions about the, obviously, each of these fields has its own distinct history, but agricultural research in particular has quite a long history of people trying to optimize crops. Yeah. So how do you feel that we're making like large improvements now as a result of these new technologies, or are these more incremental? And do you feel that they're the sorts of things that are going to help across Australia, or do you think that they need to be a bit more locally specific?
Okay, so first of all, I think that in some ways the big advances aren't necessarily the newest ones. That improved imaging enables us to use reasonably modern computer vision techniques, and putting them with relatively classical machine learning, not AI in the sense of the current buzz, allows us to develop models that could be trained on images that we've already classified, ones that have high degree of a rust infection compared to those that don't, makes it possible then to process a lot of things quite quickly. So I think what we're seeing by and large is application of more and more applications of those fundamental techniques across images that are collected in different ways. The big opportunity that up to now has always been too expensive and too difficult to generalize is how we make use of hyperspectral imagery. So with perhaps 300 or 400 bands, including many that are not visible to us, and looking for all of the possible information that may be relevant to assessing different aspects of the plant's biology and responses to light, etc. And in that respect, I think, yeah, we have lots more to learn and lots of opportunities. There's certainly real challenges in applying some of these techniques consistently across a continent as large as Australia with such diverse climate and sunlight in different regions. If you've got very bright light, it does affect the reflections that you're receiving off the plant. So a lot of these things do have to be calibrated quite locally, but underneath that, the techniques can be quite common across large regions.
No worries, thanks for that. Another challenge question we've just had come in, which is a really interesting one, and I think that will be relevant to all three. So a question come through from Patricia on the chat, which was, is Australia considered a provider of new technologies in these spaces, or a user of existing available technologies? Now, I imagine all these are globally evolving disciplines, right? Sophie, perhaps, would you like to comment?
Good question. I would say it varies depending on the omics in particular. So there might be parts where we are more of a follower than the forefront. I mean, we're in the forefront of a lot, but using the technologies that are developed. That said, I think it's a little bit of a mix. And there's definitely a lot of development, in particular in some of the synthetic biology as well, and a lot of those spaces. Proteomics obviously has been fairly, was coined by an Australian person, so that is definitely coming from here. It's sort of the world is evolving, and the research world is evolving so fast that it's always, I think, a bit of a tug and pull, and so just a bit of exchange. So we're definitely having a nice place in the field, which is nice to see with some of the investment that have been coming. So we're definitely not only behind. In a lot of places, we see that we are the same, if not sometime ahead of some of the other places worldwide.
Nice to be part of a global community then in this space. Diana, do you find the same in the imaging space?
Yeah, absolutely. I mean, NIF is trying to get cutting edge instruments and technology and trying to be the first to bring it over to the Australian researchers in terms of our total body PET systems that were recently installed, and these large field of view CT scanners. This is a technology that is it's not found, or it's not spread quite widely across Australia, and it's a technology that our researchers are very keen and honored to have and access to, basically.
I guess another aspect to this, though, is not just the technologies themselves and whether we're keeping up, but their applications and the insights that are being derived from them. Alistair, you mentioned that that from your research that there's a phenomenal diversity of the taxa you're looking at that is uniquely Australian. Do you think that has the potential to lead to new medical breakthroughs, for example?
So do Australian taxa have undiscovered properties in terms of? Yeah, absolutely, for sure. So Insulosity subaeruginosa, let's say you eat the wrong genotype, then you can be overcome with a paralysis that lasts temporarily. So some users in the community have found this phenomenon called woodlubber's paralysis. And so during their psilocybin experience, they will be totally paralyzed. They can't move or speak. And this compound, which is produced by some genotypes, could bind to peripheral serotonin receptors and cause this kind of a paralysis. we don't even know what it is. We don't even know what genotypes do it. So it's kind of like the secondary metabolites present in fungi. We barely scratched the surface of what's there. And it's criminal that we're seeing them disappear before our eyes before we even know what they can do.
Yeah, an interesting aspect of the biodiversity story there actually is the potential then that you're losing future pharmaceuticals before you can understand them thoroughly. I guess also a cautionary tale that for anyone watching along at home, I'd suggest that if you do see mushrooms and are interested in categorizing them for citizen science, perhaps just take a picture and leave it something like iNaturalist. Lovely. Well, look, I think we do have a few more minutes, but I think that covers all the major questions that we wanted to ask, unless anyone wants to add anything that they feel has been missed.
I'll just make a comment on the question you asked before, because
Sure.
I do think Australia has been a leader in this area of plant phenotyping, and one of the advantages of being part of an international community is that investments in one region can lead to a lot of innovation. Maybe that region then goes into a little bit of hibernation for a while and other regions pick it up. And certainly when APPF was started and identified at the time as a very much as a sister infrastructure to the ALA, both being biological, the work that was carried out then, particularly, well, by all three of the partners, then it was CSIRO, ANU, and the University of Adelaide, was, I think, groundbreaking to the point that most European partners that I've met with since have basically said they learned what to do from what was going on in Australia at that point. And I think we're now sort of ramping up to become a much bigger network, and I think it's our opportunity, hopefully, to start contributing again. But we're now learning a lot from how the Europeans in particular have taken some of those ideas and applied them over the last 5 to 10 years. And I certainly hope that by developing potentially cheaper and cheaper tools using simple cameras in Internet of Things solutions, we may be in a position also to produce methods that can more readily be applied in the Southern Hemisphere. So we share some characteristics with across our wide climatic range with a lot of other regions that perhaps may be able to look to us for ideas more readily than they can to Northern Europe.
That's great. Thank you, Donald. Much appreciated. And thanks everyone for taking the time today to put those presentations together. An extremely diverse set of talks, perhaps the most diverse that we've done in a single location during this webinar series. To everyone watching it along at home, feel free to fill in the poll if you haven't done so already. This talk will be available on YouTube in due course, so feel free to go back and watch it again should you wish. And in the meantime, thanks very much for joining, and we'll see you again for the next webinar in the spring.
Thanks very much.
Thank you. Bye.